See those sugar molecules across the top of the screen and that cholesterol molecule in the lower left? Those are all organic molecules. Part 2 of our chemistry lecture will focus on organic compounds. So molecules are organic if they contain carbon and hydrogen. So carbon alone doesn't do it. It has to be the combination of carbon and hydrogen. When we look at cells, there are 4 flavors of organic compounds. That is to say, there are 4 classes of organic molecules that make up the structure of cells: carbohydrates, lipids, proteins, and nucleic acids. We're going to talk about each in turn. Our first class would be the carbohydrates. Carbohydrates are made almost exclusively out of carbon, hydrogen, and oxygen. And there's a very particular ratio of those 3 elements. For every atom of carbon, there is 2 atoms of hydrogen and 1 oxygen atom. So it's as if there is a water molecule, H2O, attached to each carbon. And that's where we get the name carbohydrate. Carbohydrates have many functions. A primary function is as a source of cellular energy. Muscle cells, nerve cells can readily use carbohydrates as fuel for metabolism. The simplest form that carbohydrates come in are called monosaccharides, which literally means single sugar. So they're called simple sugars or single sugars. Glucose, that's our blood sugar. Fructose, that is the sugar in fruit. Galactose is less well known. Deoxyribose, that's the D in DNA. Sugar is actually part of DNA structure. In organic chemistry, there are lots of isomers. Isomers would be molecules with the same formula that have different structural arrangements. So glucose can exist in different forms. If you shift the oxygen that has the double bond to the carbon from the number 1 to the number 2 position, you turn glucose into fructose. So you can see they're both C6H12O6. So galactose is also an isomer of glucose. So especially the 6 carbon monosaccharides, structurally they're all very closely related to each other. In carbohydrates, the next level of complexity above a monosaccharide is a disaccharide, or a double sugar. So this forms when there's a bond between 2 monosaccharides. If we had a glucose and a fructose bonded together, that would make sucrose, or table sugar. Lactose is glucose and galactose. 2 glucose molecules bonded forms malt sugar, or maltose. So again, sucrose, that's table sugar. If you are lactose intolerant, your digestive system does not make enough of the enzyme that is capable of breaking that bond between the 2 monosaccharides in lactose. So if we continue up the hierarchy of organization in carbohydrates, we have oligosaccharides. Oligo means a few, more than 2, but not too many. So an oligosaccharide would be a short chain of sugars. This is a picture showing the membrane, the plasma membrane of a cell. And there are lots of oligosaccharides attached to our membranes that are important in cellular recognition and other important functions. The largest of the carbohydrates are the polysaccharides, and these are polymers. A polymer is a chain created by repeating molecules over and over. So polysaccharides are big, like it can be hundreds of sugars long. We have glycogen as a major polysaccharide. It is a branching chain of glucose. So we make glycogen in our muscles and in our liver. And it is there for energy. Your muscle cells will break down glycogen in glucose and use that. The glycogen in your liver is used to maintain your blood sugar levels. When you have excess glucose, you form glycogen. When you're low in blood sugar, you will break that glycogen down into glucose, right? So in this figure, A shows the formation of a disaccharide. B shows us a little piece of a very big chain called glycogen. Lipids are a second separate category of organic molecules and cells. Lipids share the characteristic of being hydrophobic, which I guess literally means afraid of water. So what that means, of course, is that they're not water soluble. They don't dissolve in water, right? They don't mix easily with water. Some of the most important lipids have molecules called fatty acids. And here are two examples of fatty acids. These are long carbon chains with lots of hydrogens attached to the carbon. And on the left-hand end, there is a group, a cluster of atoms called the carboxylic acid group. And that is where we get the name fatty acid from. The palmitic acid is said to be a saturated fatty acid because it has as many carbons as it can possibly hold. The linoleic acid below in frame B is unsaturated because in several places there is 2 bonds between carbons, and each carbon atom can only make 4 bonds. So for each double bond, we lose a hydrogen. Lipids are high in caloric content. They're worth a lot of energy because there's so many carbon and hydrogens there. When we break down an organic molecule and use it for cell energy, the real energy comes from stripping hydrogens away from carbons, and check out how many there are on offer in a fatty acid. This shows us how a molecule like a fat or oil is built. Fats and oils are lipids. They're said to be triglycerides because there are 3 fatty acids, that's the tri, bonded to one molecule called glycerol. So that 3-carbon molecule that highlighted in pink is glycerol. The 3 purple molecules are fatty acids. So adding 3 fatty acids to 1 glycerol makes a fat or oil or a triglyceride molecule. Other important lipids include phospholipids. Phospholipids are really modified triglycerides. If we take away one of the fatty acids and replace it with a phosphorus-containing cluster called a phosphate group, we have a phospholipid molecule. Phospholipids are unusual among lipids because the phosphorus-containing head region is actually hydrophilic. It actually is water-soluble, water-loving, but the tails, the fatty acid part, is hydrophobic like other lipids. So here we have this so-called amphipathic or amphiphilic molecule that has both water-loving and hydrophobic regions. We'll see lots more about phospholipids when we talk about cell membranes. That's basically what they're made out of, phospholipids. Steroids are an important group of lipids. These are very flat molecules made out of interlocking rings of hydrogen and carbon, right? So the ring's made out of carbon, has hydrogen attached to it. Cholesterol is a steroid, and cholesterol is the metabolic precursor to other steroids like the sex hormones estrogen and testosterone. So we see in this figure how cholesterol can be used to create bile salt or testosterone or estrogen by tweaking that basic molecule. All right, so our third group. This is a really big group, a really important group. That's proteins. Proteins are huge molecules. They are polymers, just like glycogen is. They're very long chains of repeating units called amino acids. That chain of amino acids would be called a polypeptide. And a polypeptide is folded into a specific shape to create a protein. The name peptide comes from the particular kind of bond that forms between two amino acids. So at the carboxylic acid end of one amino acid and the amino, the nitrogen containing end of its neighbor, a bond is formed called a peptide bond that holds the amino acids in a chain. So humans and most other organisms use a palette of 20 different amino acids to build our proteins. And the chemical and physical properties of a particular protein is dependent on the chemical and physical properties of the amino acids used to build that protein. So we see here just some examples of a few different amino acids. The first picture at the top is a generic amino acid. Then we see the simplest kind of amino acid is called glycine. And then we see examples of some other amino acids. So proteins have layers or levels of structure. And we can talk about 4 levels of protein structure. The primary structure of a protein is described as the amino acid sequence of the polypeptide. So by that we mean which of the 20 amino acids are used and in what order are they strung together to make a protein. So with 20 amino acids to choose from, and most proteins being at least 100 amino acids long, there's a lot of different primary structures. All you have to do is change one amino acid in that chain, and you've created a different primary structure. And that protein might have very different properties. Now, if you think about a chain of amino acids and you twist that chain into a spiral, or you fold that chain back on itself and create a flat, crinkly sheet, you are giving that chain of amino acids secondary structure. Right? So an alpha helix or a pleated sheet, those are examples of secondary structures. It's kind of the initial orientation that the chain takes in space. The third level of protein structure is called the tertiary structure. That is the way the entire chain is then folded into a three-dimensional shape. Here's an analogy for you. You've played with a slinky before, right? So you've got this coiled wire. That would be, in my analogy, an amino acid chain, a polypeptide that was helical. And then you tied that slinky in a knot. You would be giving it a third level of structure. You would be creating a very specific three-dimensional folding pattern. For proteins, this is really where they become functional. The shape of the protein determines what its job is, determines what its function is. Now, proteins can be made of multiple polypeptide subunits. So if you have two or more polypeptide chains bonded together, you have a protein with fourth level or quaternary structure. Imagine we took two slinkies tied in knots and crammed them together. That big mess all together would be an example of quaternary structure. This figure is walking us through the levels of protein structure. We start off with a string of amino acids, so it's sort of portrayed like a chain of beads. Every bead is an amino acid. They're held together by peptide bonds. If you know which amino acid is in each of those positions, you know the primary structure of that protein. Now, along that chain, parts of the string will fold back on itself to make crinkly sheets. Part will twist into a spiral to create an alpha helix. Those are secondary structures. In frame C, we see how the whole molecule then is folded into a specific shape. It has tertiary structure, and in this case, there are four separate polypeptides joined together to create a huge molecule that has quaternary structure. This is a hemoglobin molecule. Hemoglobin is the oxygen-binding protein in your red blood cells. It is made of four separate polypeptide subunits, right? So it has quaternary structure. It's called a tetramer. Other proteins can be made of just two subunits. That would be called a dimer. At the center of each polypeptide subunit, there's an iron-containing heme unit. That's the thing that oxygen binds to. So hemoglobin is a pretty good example of a large protein that has that fourth level of structure, that quaternary structure. When we look at examples of protein in cells and in bodies, we can often divide them into two structural categories. Some are long and skinny like a fiber. Those are called fibrous proteins. In our study of tissues and the skin, we'll see some fibrous proteins, long and thin, keratin, elastin, and collagen. Those are all tough proteins. They're very common in skin and hair and nails and connective tissues, and they're fibrous. Other proteins have very complicated three-dimensional shapes. Their tertiary structure is really elaborate, so they're folded into a glob. They're globular proteins, more compact, more spherical, more complicated tertiary and quaternary structures. These tend to be really important in physiology. Antibodies, and hormones and enzymes. Those are proteins that have complicated globular structure if they are mistreated through heat or pH or chemical insults or age. The proteins can become unfolded, they can lose their structure, and they become non-functional. Our fourth group of organic molecules are the nucleic acids. Their common unit molecule is called the nucleotide. So nucleotides are to nucleic acids what amino acids are to proteins, or what monosaccharides are to carbohydrates. Nucleotides come in basically 5 flavors. There's a nucleotide called adenine, one called thymine, cytosine, guanine, and uracil. So when we see large nucleic acids, they are made out of combinations of these 5 different types of nucleotides that we refer to by a 1-letter abbreviation. This is a structure of the basic structure of a nucleotide. So at the bottom, we see that a nucleotide has a 5-carbon sugar as part of its structure, and that sugar is of 2 types, either called deoxyribose or ribose. Sugars commonly end in OSE suffix. So the nucleotide in figure C is one that uses deoxyribose as the sugar. It has a phosphorus-containing group attached to it, just like phospholipids did, that helps make nucleotides water-soluble. And then there's a variable part called the nitrogen base, and that's where we get those 5 different kinds of nucleotides, A, T, G, C, and U. Those are the nitrogen bases. This is a picture of what those nitrogen bases look like. So just as the 20 amino acids can have different groups attached to them, our different nucleotides have these bases attached to them. Probably the most famous nucleic acid is called DNA. The Na stands for nucleic acid, and the D stands for deoxyribose, the sugar deoxyribose. So DNA is our genetic material. It consists of 2 chains of nucleotides. They're twisted into a helix, and you can find it in the nucleus of the cell. That's what your chromosomes are made out of. That is our genetic material. That is the universal genetic material in living organisms. Before a cell divides, it will copy its DNA. We call it replication, so that both of the daughter cells will inherit a full complement of the genetic instructions. So our cells are very good at accurately copying our DNA, which is made out of billions of nucleotides. DNA serves as our genetic material in large part by providing the directions to cells for how to make proteins. They provide the instructions for how to synthesize proteins. DNA will tell a cell which amino acids should be strung together in what order to make a particular protein. This is a short segment of a DNA molecule. We see there are 2 chains of nucleotides. The whole molecule is twisted into a helical shape. The sequence of A and T and G and C bases along the molecule is how DNA codes different messages. This is an interesting figure here. We see kind of a naked DNA molecule on the left. To fit into our cells, DNA has to be coiled up, wrapped around proteins that compacts it into a state called chromatin. And when it's time to divide, that chromatin gets even further compact into very short, dense bodies called chromosomes. And when you see an image of a chromosome, it will typically have 2 equal parts to it, and those represent the 2 copies of the DNA, that replication that happened prior to division. This is a cartoony figure that gives us a brief glimpse into how DNA works. So what we're seeing in this figure is at the top is the nucleus of the cell, and the DNA is that red molecule. And one segment of DNA that codes for a protein is a gene. And the genes do not leave the nucleus, yet they provide the directions for how to make proteins. The work of building a protein happens out in the cytoplasm using structures called ribosomes. So to get the information from the nucleus to the ribosome, we make a temporary disposable copy of the gene using an RNA molecule. We create an RNA copy of the DNA. RNA is a single-stranded nucleic acid chain that uses the ribose sugar. So that RNA molecule, which is yellow in this figure, can exit the nucleus and complex with a ribosome. That's the thing that sort of looks like a purple popcorn, and coordinate the building of proteins. So we see the individual green squares there represent amino acids, and they're being strung together in the right order, dictated by the sequence of nucleotides in the RNA, which of course was based on the DNA. Here's one more really important nucleic acid. It's called ATP, and that initialism stands for adenosine triphosphate. So unlike DNA and RNA, ATP is not a chain. It is a single nucleotide that has 3 phosphate groups attached to it. And that molecule is the most immediately usable source of energy for cells. So your blood sugar is really important for cell energy, right? But a glucose molecule is an inconveniently large amount of energy. So through the process of cell respiration, we break molecules like glucose down, release the energy that's inherent in its bonds, and use that energy to make ATPs that we can spend one at a time. Each ATP represents a small amount of energy that's available immediately to do work. So here is an ATP molecule, and we see there are 3 phosphate groups attached to a ribose sugar, and it has the nitrogen base is the A variety, adenine. We spend an ATP by breaking off that last phosphate group. That releases energy, and what you have left is a nucleotide that has 2 phosphate groups that is called ADP, adenosine diphosphate. And later, when you have energy available, you would tack that phosphate group back on and resynthesize that molecule for use again. So this figure shows us how ATP drives cellular work. On the left, that molecule of ATP represents stored energy. By breaking one of those phosphate groups off, you are releasing that energy. You can use that to make proteins change shape. You can use that to make muscles contract. You can use that to drive a chemical reaction that requires an input of energy, like the building of a protein. Okay, I think I'll stop here. We got one more bit of chemistry to talk about enzymes. That'll be a shorter presentation, but we'll make that our third and last chemistry lecture.